Hello, I am Matthew Habimbade, and I'm excited to walk you through the study on a really important topic, which is cessation of CCL2 inhibition accelerates breast cancer metastasis by promoting angiogenesis. So we'll be driving into how stopping the inhibition of a specific chemokine called CCL2 can actually make breast cancer spread faster by promoting the growth of a new blood vessel. And moving forward, looking at the next slide, which is slide 3, we will see that the discontinuation of anti-CCL2 treatments increases lung metastasis and accelerated the death of the mice. So here we can see that the stopping the CCL2 inhibition led to a sharp decline in the survival. When we look at the first graph, which was showing the Kaplan-Meier curve, it showed that the mice actually lived longer with continuous treatment. But once the moment we stopped the treatment, lung metastasis rapidly increases, as shown in the plots actually stated here. And this underscores the risk of stopping CCL2 inhibition. And when we move to the next slide, we'll see that the monocyte actually induced blood vessel formation and the proliferation of lung metastasis after discontinuation of anti-CCL2 treatment. Okay, we eventually found that monocytes after CCL2 inhibition actually stopped and contributed majorly to the formation of blood vessels, and it definitely increased the tumor cell growth in the lungs. So, and the images above actually show that the image on the bottom left actually shows that the big increase in CD31 positive blood vessel in lung metastasis indicates that angiogenesis or blood vessel formation is ramping up, and is ramping up on the top right. We also have K167 staining, which highlights the proliferating cells with this metastasis. So what's happening here is that there is a surge in tumor cell proliferation after treatment stops, and together the images actually make the one on the right and the one on the left actually makes it clear that monocytes are driving both the formation of the new blood vessel and the growth of the cancer cells in the lung after we stop until CCL2 therapy. So when we look at the fifth slide was talking about the monocytes, which were the monocyte definitely induced the blood vessel formation and proliferation of lung metastasis after discontinuation of the CCL2 treatment. So continuing with this idea, we saw that after stopping CCL2 inhibition, both the metastatic area and the number of proliferating cells significantly increased. So, which actually reinforces how critical monocytes are in driving growth when the treatment was altered. So, eventually, when we look at that very well, we will see that it was an aggressive growth that was actually noticed. And on the slide 6, the IL-6 treatment after cessation of anti-CCL2 therapy prevent the overshoot of lung metastasis. So we then tested combining anti-IL-6 with anti-CCL6 therapy, and the results were promising. And blocking IL-6 after stopping CCL2 inhibition is significantly reduced the surge in lung metastasis, according to the figures here. So the images here actually shows that when we lower FA level and fewer CD31 positive cell, which suggests that combination of both could prevent the excessive tumor growth. That is what we observed in the images. And when we look at the seventh slide was talking about the combined anti-CCL2 and VEGF treatment. It also reduces the angiogenesis and the tumor cell proliferation in lung metastasis. So the CD31 positive blood vessel indicates that angiogenesis is reduced on the right. So the number of K167 positive cell is also very lower, which means that tumor cell proliferation is being effectively controlled. So this results suggest that using anti-CCL2 alongside VEGF could be a powerful strategy to limit the spread and growth of metastatic tumors. So finally, the 8th slide actually emphasizes on the benefit of combining CCL2 with the VEGF treatment. So the image here actually shows the combination reduces the level of VEGF-A lung tissue and lower p-stat levels, both of which are associated with reduced angiogenesis and tumor growth. The left image actually gave us an insight into the markers like VEGF and p-stat3, this which eventually supports the idea that targeting multiple pathways could be a key to effectively prevent the progression of metastatic breast cancer. So in conclusion, our study actually reveals that complexities in targeting tumor microenvironments in metastatic breast cancer. While anti-CCL2 therapy reduces metastatic initially, discontinuing the treatment can lead to a rebound effect. So characterized by increased monocyte infiltration, elevated IL-6 level, and enhanced angiogenesis in metastatic site. So this emphasizes the importance of continuous treatment and the potential for combination therapies to effectively manage cancer therapies.